I'm Raghavendran, bioinformatics mentor with Fine Biotech, and I welcome you all to this overview session on the Summer 22 OmicsLogic training programs. So this part of the session will give you more detail and additional details about the program design, about the curriculum, and about resources. So as you know, this summer we have a range of programs that you can look into further and register so that you can learn a little bit more about bioinformatics and how it is applied to various different uh, topics related to biology and bioinformatics. So this program will generally be offered virtually around the topics of infectious diseases. It also includes coding in R and in Python. It also includes bioinformatics analysis of gene expression data in transcriptomics, and in general, introduction to big data bioinformatics. So this program will actually help you to complete a research. At the end of this, we also have a research fellowship program, and Dr. Mohit will talk more about this research fellowship program. So this research fellowship program is specifically designed to help you complete a project leveraging public domain data from using big data infrastructure and exploiting training and support from all of us, from our team and our mentors. So let's go to a little bit more detail about these programs one by one and understand what they offer to students and faculties. So we start with introduction to bioinformatics, where we actually introduce you to the big world of bioinformatics and big data.
Sorry for interruption. Hello. Can you hear me?
Sonalika, can you mute? Yes, thank you very much.
Yes, sir, you can proceed further. Shivam, can you put your query in the chat, please? We'll answer your query towards the end of the session. Thank you so much. Over to you, sir.
Yes, thank you very much. Please, please put your queries and enter your queries in the chat. I'm following chat as I'm chatting with you. So, so let me share again. Okay. So the Introduction to Bioinformatics program is designed to introduce you to the world of big data biology and data science tools. So it starts with the basics of next generation sequencing, and it starts with what are the different omics data types that you will be learning, and what are the different omics data types that are used generally to understand the biology and different biology of the system from molecular level. It also extends to what are the analysis methods and options that we have to use for or to employ on different omics data types, so that we can extract the maximum information from these data types. Okay, somebody says that my voice is so feeble. Let me sound up. Thank you very much. So in the end, by the end of this big data bioinformatics program, you'll also get an introduction to how to use or how to apply advanced statistical methods like machine learning and deep learning to the structured data that we generate from this next generation sequencing data. So in the end, you will achieve all of your achievements will be summarized in certificates that you can share with your peers. So yeah. This you went through. We have several extensive training materials prepared for you, like associated courses, lessons from OmicsLogic platform, asynchronous videos, live hands-on demo sessions, and we also have prepared curated example projects, and which takes a publication and the data associated with the publication to an analysis method of its own, and walks you through the whole process of from the data set through through specifically designed analysis methods for the data set and biological interpretation and conclusion and comparison with the original findings. And we also have in the research fellowship program that you will hear today. So we promote project-based learning and interactive sessions for exploring bioinformatics as an option. So we also have several lessons which are coding lessons that that is structured in an interactive fashion so that you can explore coding for bioinformatics. So the analysis methods that we have discussed or that we have in hand for different omics data are usually applied or employed in two approaches. So one of the approach is for code independent approach is for those who does not want to venture into the world of coding and who is not very comfortable in coding. So these methods, these code independent infrastructure methods will include hands-on exercises in our big data bioinfo platform like T-Bioinfo platform. And this provides a simple and intuitive user interface for processing, analyzing and digital data and different structured data also. So we can employ all different kinds of analysis methods. Here you will get a 5 terabyte storage so that you can store your data that usually is huge when you involve big data bioinformatics, and you will get support from us, and you'll also get all the necessary associated resources and lessons and videos so that you can set up your analysis pipeline independently and vary the methods in the analysis pipeline to suit your data. So this analysis pipelines include both the analysis of next generation sequencing data to arrive at a structured table, to structured data or a structured list of variants. And then from there, it also includes the downstream analysis of whatever you have generated, the structured data that you have generated, so that the downstream analysis applies various statistical methods, machine learning methods, so that you can infer and explore the meaningful relationships that is buried deep within your data. So here is an example of the code independent pipeline that I was talking about, and this pipeline builder interface in the T-Bioinfo platform is color coded so that you can establish a connection between a logical connection between how your analysis methods are progressing. And the data input is an input file, and the analysis can be chosen based on the type of research and the type of question that you are going to ask. We usually have 3 basic colors that separates the analysis methods. Blue is for pre-processing, red is for mapping, and green is for quantification. And you can explore orange is for downstream analysis. The interactive output, we also have option for interactive output, which allows for easy visualization of the data outputs. And there are several demo pipelines which will give you an experience of setting up an original pipeline and downloading the output, associated output, to get a feel for how to set up big data bioinfo informatics pipeline for next generation sequencing data and for downstream analysis, and what are the different kind of outputs that you will get and how you can make sense of this output in the connotation with the biological relevance or phenotypical association of the samples. So this is part of the interactive output. So for those who want to expand your skill sets to coding and venture into learning coding along with their analysis, we have lessons that involve coding, and these lessons are divided into several steps. So the first step is always to understand the context or the aim of the analysis, and it is followed with the logic behind the analysis that we are performing. And then we have a learn the code section. Learn the code section, where each and every parts of this code are explained to you in a small piece by piece manner. And finally, you'll have practice the code section, where you can practice whatever you have learned. And then in the end, you will have option to download, or you'll have option to access the full code document in a Jupyter notebook or a Google Colab notebook format, so that you can independently modify the code to suit your analysis or data requirements. So each session is actually planned for an hour-long video, and you will have more associated resources that you will have to go through. And I think our Sonalika will actually talk more about how much you should spend for each and every one of these sessions, so that you can gain or get maximum information from these sessions or resources from these sessions. So yeah. So for our next program, we have biomedical data analysis using R and Python. So the introduction to bioinformatics section will introduce you to the different analysis methods. But if you want to delve deep into the world of data analysis and data science of omics data or big omics data, then you have to choose an option like this. So in this program, for example, the focus is on learning data science and not programming. So programming we use as a tool to learn data science. The more focus and emphasis is on understanding the logic behind the methods and tweaking certain part of the programming or of the code blocks so that it suits your data or your analysis methods. So yeah. So why do we choose only Python and R? So we know that Python and R are the major resources that is used for bioinformatics data analysis. And I don't want to create any war between people who like Python and people who like R. So we are on the fence, or we are of an opinion that it is not a good idea as a bioinformatics enthusiast or of a data science enthusiast to pigeonhole yourself into one of these methods. Both have their advantage and both have their shortcomings. So you should learn enough concepts from both the tools as much as you can, so that you can apply either one of them depending upon the type of data, depending upon the type of analysis that you use. And that is what we also promote. For most of the lessons that you have, we have all these analysis methods divided into 3 parts. The first part, as I actually mentioned earlier, it's code independent block, where you can get the experience of performing these analysis using the big data bioinfo platform. And the second portion is running the same analysis in an environment like R. And the third one is using environment like Python. So you will have complementary information from all these 3 different resources. So we first start with the basics of getting started with R and Python, where we start absolutely from the basic, like how to load a data into R's environment, or how to load a file into Python's environment, which is absolutely the most necessary skill that you want to have if you want to use R and Python for bioinformatics data analysis. And then we up the ante with trying to analyze or trying to read DNA sequences, and how to compare DNA sequences in R and Python, and how to take your first steps into bioinformatics data analysis. So all of the data that is discussed and all of the data that you are going to load are from either of the omics data, specifically prepared and specially prepared for you to run all these analysis in your in the interactive platform that you will learn, or in your R studio that you have locally installed, or Python environment that you have locally installed in your computer. And we and then to increase the applications of R and Python into the world of data science, we have several different data processing and visualization and advanced visualization methods that you will get hands-on to, and how different visualization is also going to help you learn about the patterns that you can generally observe from the data. It's just not you run this code, boom, you get a scatter plot. If you run this code, you get a histogram. But more emphasis is on what are you going to learn from histogram, and why is histogram a better option when compared to scatter plot. So more of these kind of discussion is what we will be having in this one. So to start from absolute basic, you always have to start from somewhere. So we have to give you a detailed introduction in the first lesson, or introduction to bioinformatic analysis in R, about some of the concepts that you will mostly use, or you will use frequently in your data analysis, like what are the different objects like data frame and the array and matrix that you will be using for your omics data analysis. What are the different basic functions, like how to find the dimension of the array, how to find the size of some of the elements, and how to find the how to index or take specific portions from the data frame, or how to restructure data frame based on your requirements. So all of these would be covered extensively up at the start, and then as you progress in the program, so these will be used repetitively, so that you can get a feeling for how these are used in the omics data analysis. So you can say that we in Python or in our world, we actually quickly introduce you after your data exploration and data loading and transforming. We quickly introduce you to advanced statistical methods like machine learning and with supervised and unsupervised machine learning, and comparing different supervised classification algorithm and etc. So this is in BioML course in Python, which and also available in R, which compares different machine learning methods for the same or similar data, so that you can clearly get an understanding of how same or similar analysis methods interpret the pattern that is that you can find, or that is available in the data, that is present in the data. So always you start with load and transform and start exploring the data, and then before going into further analysis methods. So all of these methods will actually take the structured data that I was talking to you about. So this is a table output, tabular output, that you can generally get access to from publicly available data set, or if you have next generation sequencing data, you can get to this structured data from running one of those pipelines that we have discussed before, and you will also see one more example of that. So the practical coding assignments in our in the portal, on portal, is designed in such a way that it will help you to understand the different elements of code R from coding Python. For example, this is an example of interactive console that we are going to offer you for both Python and R environment, where you will have your code, where you'll have the code that you can write on the console, and then the console will and the right portion of the console will be where it is being executed, and interactive output is going to be printed out. And you will also have, if you have codes to prepare, or if you have codes to print out plots, and the plots will also appear below. So this is embedded into each and every one of these coding lessons, so that whatever you have achieved in learning the code section, you can apply it into this practice the code area. So and we also have some more test your ability to fill up the codes, where we modify some of the coding codes, so that you can complete those syntaxes, and we deliberately leave out some parts of the code, so that you can access yourself, or you can try to complete those codes, so, and learn the code section area. So once you are through with the practice section, where you have completed these codes successfully, and these practice sections are designed in such a way to increase the level of difficulties, and once you are through, you will be given either an option to perform an assignment or complete an assignment, or you'll be given access to Google Collaboratory Notebook, or a Jupyter Notebook kind of link, where you can get all the codes that are discussed in the lesson, and a little bit more other options also. So you can get a feeling for running the code either one by one, or in unison from start to end. And you also, since you have the access, you also have the freedom to play with the code, to change the analysis methods, to compare one method with the other one, and etc. So as I mentioned at the start, we don't focus on making you learn programming. We don't focus on making you learn programming, but we let you understand the data science aspect of the programming. So how different strategies of these analysis work together, and how by employing different data processing methods will help you define the hypothesis, will help you refine the hypothesis, and will help you prepare the data for meaningful applications. Most of the time in the data science application method, most of the time spent in this portion and this portion. The data analysis and summary of statistics will take only will take limited amount of your time and limited amount of your coding areas. So go back, I think, yeah. So most of the time you're going to spend in processing, cleaning, and pre-processing the input data such that it fits the analysis requirements of it fits the statistical requirements of different analysis methods. And data mining and visualization and exploratory analysis perform very important part, key portion in refining your hypothesis. You look for pattern. If that pattern is present, then you refine your hypothesis. Or if some of the initial early exploratory visualization provides you with a different different idea or a different view, so you expand on that view, you go in that direction. That's the power of data science. We don't start with the fixed hypothesis. We explore the data for available hypothesis, and then we keep refining the hypothesis so that our data can explore the relationships in the features or in the genes or in different aspects of different portions of the data to reject or accept or to evaluate the hypothesis. So I have a question where, before going next, when using code written by someone else, there are concerns about copyright issues and whether it is necessary to get coders' permission to copyright. So if you are getting the code, or if you're downloading the code from the coder's computer without their knowledge, then of course you have to get permission. But there are codes which are left for you to explore in the freely available resources like GitHub or in Google Collaboratory. So in these portions, you can just mention modified from this code so that you are being a good person and then acknowledging from where the code source is being taken from. And most of the publication nowadays require the authors to give link to their code so that people can reproduce their results. So once they give permission, or once they upload the code in downloadable public resources and repositories like Google Collaboratory, Google Colabs and in GitHub, then it is free for you to take, provided that you acknowledge them accordingly. So whenever you want to take codes written by somebody, please acknowledge them. This is a code that is taken from this link, and it is modified to suit this specific concern, this specific analysis method. Okay. So the difference between, for example, choosing a specific omics data analysis track and choosing, I went back again, sorry. Choosing specific omics data analysis track and program and choosing data science programming, data science track is that in the data science track, you will get, you will have experience in dealing with the structured data that is generated from variety of different omics data analysis. You can get either transcriptomics data, you can get a list of variants in the genomics data, and you can get abundance table from the metagenomics data. Each one of these structured data that you can get your hands on requires different concoction or different combinations of programming methods, and so that you can explore the variety of patterns that is associated in the data, that is buried deep in the data. So this can be as simple as, the analysis can be as simple as performing a t-test in Excel, or as complex as performing hierarchical clustering or unsupervised clustering, and then followed by supervised classification for different one of these clusters. So it can go from very simple methods to even complex and complicated